Chapter 18, and in Chapter 18 we cover the immune system, and these are the topics that we'll be covering. We will start off with the innate immune system. The innate immune system is also known as the non-specific or general immune response, which means that the body will respond to anything invading the body regardless of what that invader is or what that pathogen is. Whether it's a bacteria, a virus, a fungus, a parasite, it doesn't really matter. These are all general, non-specific responses that the body will initiate in order to get rid of that pathogen. And those involve your defenses at the body surfaces. And these are considered your first barrier, your barriers to prevent pathogens from invading the body. And then you have inflammation, interferons, fever, phagocytosis, and the complement system. So we'll talk about them individually. The defenses at the body surfaces, these are your first lines of defense. So the best way to defend your body would be to prevent things from invading to begin with. So for example, a mechanical barrier, for example, would be to have intact skin and mucous membrane. And really the key word here is intact. So as soon as we get a wound, we have lost that mechanical barrier. Other barriers, for example, could be chemical barriers, like the tears of the eyes or the hydrochloric acid in the gastric juice. We also have different enzymes in our sweat. All of these are different examples of chemical barriers. And here you can see an example where we have that mechanical barrier. So there is an interruption in the skin. For example, here you can see a splinter with bacteria being introduced into the skin. And down here you see the capillaries with the neutrophils and RBCs. These tissues that are now damaged are going to release different chemicals. These chemicals are going to induce dilation of these capillaries, and they are going to open up these pores of the capillaries. And that is going to lead to the neutrophils being able to squeeze themselves out of these little tiny pores. As you can see here, that is a process known as diapedesis, where it comes from the name pedal, kind of like these neutrophils having feet-like extensions and able to squeeze themselves out of these capillary pores. Remember, neutrophils are our first responders, so these are the first cells to get to the area of damage. They are going to eat up the bacteria through phagocytosis, and you can see here the capillary will go back to normal, the pores will shut down again, and the fibroblasts that are found in skin are going to start the healing process. Now, what are some of the inflammatory mediators that mediate inflammation? We've got, for example, there's a whole list, but I want you to kind of focus on 3 of them: complement, histamine, and cytokines. So complement, and we're going to talk about that in detail in a couple of slides. These are a whole bunch of plasma proteins that once you activate one, you are activating the whole system. And this is kind of like the immune system on steroids, where it does so many different things. It kills pathogens directly, or it can flag down pathogens, kind of like putting a red flag on the pathogen, telling the rest of the immune system, alerting them that this is an unusual, or this is an infected cell, or it is a pathogen. And that process of red flagging is known as opsinization. Histamine is released by mast cells and injured cells. So you've seen in that image how the histamine that was released by those injured cells are going to increase the vascular permeability. Cytokines, these are different chemicals that are secreted by immune cells, and they kind of work as paracrine hormones, where they further activate other immune cells, and they also call in other cells. And the process of calling in cells to an area of inflammation or an infection is known as chemoattraction or chemotaxis. Here you can see the process of phagocytosis, where this phagocytic cell, and remember, a lot of our cells are phagocytic cells. The most common ones, or the most phagocytic ones are the M&Ms, your macrophages, neutrophils, and monocytes. And you can see it here engulfing the microbe through endocytosis. You have now made a phagosome that has the bacteria or has the microbe. The cells, if you guys remember, our immune cells have lysosomes that are full of enzymes that can digest microbes. That lysosome will fuse with the phagosome to make a bigger phagolysosome that is going to literally digest the microbe into smaller parts and then release these products into either the cell or release them outside of the cell. So what are the functions of the phagocytic cells in the innate immune response? We kind of already talked about phagocytosis and how it's going to kill the microbes. And it will do a couple of other things. It can regulate the inflammatory response. It could cause the extracellular killing of microbes. So just like it can kill the microbes on the inside of the cell, it can also kill the microbes outside of the cell. It can lead to the activation of the clotting system. Remember when we talked in chapter 12 about clotting mechanisms? Sometimes what an infection does would, in order to prevent the spread of the infection, it would induce a blood clot. So on one side, you're kind of trapping the infection in this area and preventing it from spreading, but on the other hand, you've blocked the oxygen supply to the area. Okay, so that can be mediated by chemicals released by phagocytic cells. The other chemicals that come out are going to mediate the overall response of the body, so a systemic response of the body to infections. For example, getting a fever, feeling fatigued, loss of appetite, and so on. Those are responses to the body's response to some of the chemicals released by the phagocytic cells. Now, what is the complement system? Like I said, a complement system is a whole family of plasma proteins. And when you activate the first one, you pretty much go down the line, kind of like this domino effect. There are about 30 different proteins, and we name them C1 for complement 1, C2, C3, and so on. The most important of this whole cascade happens to be protein 3. So C3 is the most important. Once you've reached the activation of C3, there's really no going back. And eventually, when you're done with the whole cascade, you're going to make this huge protein known as the membrane attack complex, or the MAC. The membrane attack complex does exactly what the name indicates. It attacks the plasma membrane of bacteria, or if it was a virus, it would attack the protein coat of the virus and start making holes. It'll drill holes into that membrane. So make these pore-like channels in the membrane. Now that you've destroyed their membranes, well, they're going to leak all of their contents out. Water and ions and everything will leak into the cell. So it pretty much, damaged the cell. You've killed that cell, killed the microbe. But again, these plasma proteins are in an inactive form. You have to activate them. And there are two different ways to activate them. One of them is a classical pathway, and that needs the presence of antibodies. We will learn that we have 5 different kinds of antibodies, and the 2 antibodies that can activate the complement system is IgG and IgM. Those are the only 2 that can activate this complement system. And when we activate the system using the classical pathway, we are going to start at C1. But there's an alternative pathway where we don't need antibodies, and we're going to actually skip C1 and C2, and we'll start at C3. So I want to go to the image to kind of explain how C3 works. What C3 does is that it has a little tiny protein on it known as C3b. C3b detaches from that bigger C3 protein and attaches to the bacteria, basically flagging it. So again, a process known as opsinization. Now, phagocytic cells that have the C3b receptor are able to identify the C3b antigen that's now attached to the bacteria, and now they know that this is a pathogen and I can go ahead and eat that up or engulf it through phagocytosis. So what are the functions of your complement proteins? Well, they could either, as we mentioned, they could directly destroy the microbes through the membrane attack complex, making those little tiny holes in the membranes. We've also talked about opsinization. That's why they enhance phagocytosis, because they red flag bacteria through that C3b antigen. It can also make different chemicals to call other immune cells to come, and we called that process chemotaxis. It also leads to vasodilation and increasing the permeability of capillaries to enhance the inflammatory process. Now for interferons, and whenever you hear the word interferon, you want to think of two different things: viruses and tumor cells. Okay, so one of the the chemicals that will come out of a cell that is infected with a virus is an interferon known as interferon 1 or type 1 interferon. And basically, to understand how interferon 1 works, we need to understand how a virus can infect a cell. When a virus infects a cell, it takes over its nucleus. And instead of the nucleus being making things that the actual cell needs, the nucleus is now going to make more and more copies of that virus. And then these fresher copies are going to go out and infect more and more cells. So this infected cell basically knows that it's done and over with, and all it has to do is to notify all of the neighboring cells that, be on the lookout, I'm infected with a virus, try to protect yourselves. And it does so, the message that it sends out is type 1 interferon. So I see the type 1 interferon going to these healthy cells. Now these healthy cells get the message and they start to make what is known as an antiviral protein. So if a virus tries to infect these cells that have already been pre-warned, these antiviral proteins are going to prevent the virus from taking over their nuclei and prevent it from replicating itself. So replication is the word that we use when we talk about the multiplication of viruses. Another thing that interferon 1 does is that it kills tumor cells. And that's why there's a lot of research into why don't we use that as an agent to kill cancerous cells. And it also generates a fever, and we call that an endogenous pyrogen. Okay, so these are what your type 1 interferon does. Again, it's an endogenous pyrogen leading to fevers. It will send out signals, or it is the signal being sent out to the healthy cells to prevent the viruses from being able to replicate in them. And that's why we call an interferon, we consider it an antiviral, and it also kills tumor cells. Now that is brings us to the next topic, which is the adaptive immune system.